Welcome back to the deep dive. This is where we take some really fascinating research and break it down, looking for those big game-changing ideas.
Right.
And today we're tackling something huge in medicine, finding new antibodies. Now, antibodies are critical. I mean, almost half of new biopharma approvals involve them.
They're absolutely central.
But making them, it's always been, well, tough, slow, super expensive, and honestly, often fails.
Yeah, the traditional ways like immunizing animals or screening these massive libraries of molecules, they eat up resources, take forever, and hitting difficult targets is notoriously hard.
Exactly. The problem has really been finding that one good molecule. You might scream thousands, maybe millions,
Literally millions sometimes.
and it takes months, or like you said, even years. Plus, you don't have much control over where exactly it binds on the target molecule.
Right. That specificity is key. And for some targets, especially those with tricky shapes or what we call difficult epitopes, the standard methods they just can't reliably get there.
So a picture of massive challenge, right? Low success rates, not enough control, impossible targets. Sounds pretty daunting.
It has been for a long time.
Which brings us to today's focus, a really exciting development from the Chai Discovery team. It's a new AI system called Chai-2, and they're claiming something pretty bold, zero-shot antibody design.
Yeah, and that zero-shot part is, that's the really crucial bit. It means the AI can design these complex molecules completely from scratch.
From scratch. So without needing an existing antibody to start from.
Exactly. No starting binder needed, no lengthy tweaking for every single target. It's a fundamental shift in precision and approach.
Okay, so our mission today for you listening is to really unpack this. What is Chai-2? How is it changing molecular design? We'll look at the success rates they're reporting, which sound frankly amazing, and think about what this means for medicine's future.
It's about moving away from that trial and error guesswork
Right.
towards something more like, well, programmable molecular engineering, designing molecules intentionally.
Okay, but before we get into Chai-2 itself, let's just double-click on that problem space. Why is traditional antibody discovery so incredibly hard? What are the real bottlenecks?
Well, the biggest one, historically, has been those abysmal hit rates, especially for computational methods before this. You were often looking at success rates way below 0.1%.
0.1%. Wow. So for every 1,000 designs you try, maybe one works, if you're lucky.
If you're lucky, yeah, sometimes none. And that meant you had to rely on brute force,
Just sheer numbers.
massive experimental screens, testing thousands, millions of candidates in the lab. That's why it took so long, cost so much. It fundamentally limited what targets people could even afford to go after.
And it wasn't just finding something that stuck. Right. It had to stick in the right place to actually do something useful therapeutically.
Precisely. That lack of control over the binding site, the epitope, was a huge frustration. You might find a binder, but if it doesn't hit the functional spot on the target, well, it's not much use.
So that's why we have these so-called undruggable targets, because we just couldn't design a key precise enough for their lock.
That's a big part of it, yeah. We lacked the tools for that fine-grained, specific design.
Okay, we've set the stage. Huge hurdles. Feels like a problem tailor-made for a breakthrough. So tell us about Chai-2. How does it start to change this picture?
Right, so Chai-2 is being described as the first fully zero-shot generative platform that achieves double-digit experimental success rates for designing antibodies de novo.
De novo meaning from scratch again, right?
Exactly, from scratch.
And double-digit success rates.
Yeah.
Compared to less than 0.1%, that sounds like a massive jump. Can you put that into context?
It's a huge leap. We're talking about an average success rate of around 16% for antibodies and these smaller nanobodies. That's roughly a 100-fold improvement.
100 times better.
Yeah, a 100-fold increase over those earlier computational benchmarks. It basically means you can skip a lot of that massive upfront experimental screening that used to be necessary.
And the Chai Discovery team mentions speed, too, compressing discovery from months or years down to 2 weeks. Is that right?
2 weeks for hit discovery. It's a dramatic acceleration. Think about it. Instead of needing massive lab setups for high-throughput screening.
You could potentially find hits in a single small lab plate.
Yeah, exactly. Reliable discovery in something like a single 24-well plate becomes feasible. That lowers the cost, lowers the barrier to entry, and just speeds everything up.
Okay, so how? How does Chai-2 actually achieve this? What's the secret sauce, the technical innovation?
Well, the core is what they call a multimodal generative architecture.
Multimodal, meaning?
Meaning it integrates different kinds of data. It's not just looking at the protein sequence. Critically, it incorporates all atom structure prediction.
All atoms. So it's predicting the 3D shape down to where every single atom sits.
Precisely. It understands the precise atomic structure, the interactions, and then it can generate completely new designs that are predicted to fit a specific target, often at a specific location.
So you can tell it where you want the antibody to bind at epitope specificity.
Yes, that's a key capability, designing for specific epitopes.
That's incredible. And is it just for standard antibodies, or can it do other things too?
It's actually quite versatile. The reports show it designing standard scFv antibodies, those smaller nanobodies or VHH domains, and even things called mini-proteins, which are these really compact, stable protein structures.
And all of that zero shot, no template needed.
Correct. Zero shot, no known starting binder required, no target-specific tuning beforehand. It just designs based on the target information. You can even prompt it with multiple targets if you want something that binds to, say, different virus variants.
That's really powerful. But are the designs truly new, or are they just, slightly tweaking existing antibodies? Because novelty is important here.
That's a really good question, and they address it directly. The designs are novel and diverse. They measure this structurally. The designs are typically at least 10 angstroms RMSD away from the closest known antibody structure.
And 10 angstroms is pretty different structurally.
Oh yeah, that's a significant difference in 3D space. And in terms of sequence, they have a CDR edit distance greater than 10 from known examples. The CDRs are the key binding loops. So yes, these are genuinely new molecular shapes and sequences, not just copies.
Okay, so the theory sounds amazing. The architecture is advanced. But let's talk validation. The proof. What happened when they actually made these AI-designed molecules in the lab?
Right, this is where it gets really impressive. They ran a big test for antibodies and nanobodies. They picked 52 different protein targets.
Okay.
And crucially, none of these 52 targets had any known antibody binder in the main public database, SABDAB. So these were genuinely hard, unsolved problems for antibody design.
A clean slate challenge. How did Chai-2 do?
The results were remarkable. In just one round of experiments, testing maybe 20 designs per target, they got an average binding rate of 16%.
16%. On targets with no known binders.
Exactly. It broke down to about 20% for the nanobodies, VHHs, and nearly 14% for the scFv antibodies. And maybe even more significantly, they found at least one working binder for half of the targets. 26 out of 52.
Wow. Found a key for half of these previously unlocked doors in one shot.
Pretty much.
Yeah.
And they didn't just bind. Further tests showed these designed antibodies were stable, specific, and didn't have issues like polyreactivity, meaning they didn't just stick randomly to everything. They behaved like proper therapeutic candidates should.
That's huge for antibodies. What about those midi-proteins you mentioned? How did it do there?
Even better, actually. For mini-protein design, Chai-2 hit a 68% wet lab hit rate.
68%. That's almost 2 out of 3 designs worked.
Yeah, an astonishingly high rate. And these weren't just weak binders. They were often getting picomolar affinities.
Yeah.
That's incredibly tight binding, the kind you want for a potent drug.
Incredible. And did it manage to crack any really tough nuts, targets that were known to be difficult?
Yes, they specifically highlight TNF-alpha. That's an important drug target, but notoriously hard for computational design because its binding site is kind of flat and chemically challenging.
Okay.
Chai-2 successfully designed, apparently, the first computationally generated binder against TNF-alpha. And across the board for many proteins, they showed at least a 3-fold better hit rate than the next best computational methods.
So consistently outperforming, even on the hard stuff, these numbers are really compelling. Are there still challenges, though? Does this solve everything, or does it maybe create new bottlenecks downstream?
That's a fair point. Well, Chai-2 seems to revolutionize the design and initial discovery phase, finding those first good candidates. You still have the rest of the drug development pipeline.
Right, manufacturing, formulation, all the clinical trials.
Exactly. Those parts still take time and effort. Chai-2 gives you much better starting points, potentially much faster. But the journey to an approved drug is still long. The challenge perhaps shifts from finding a lead to efficiently developing the leads Chai-2 provides.
Makes sense. It accelerates the start but doesn't eliminate the whole race.
Okay, let's zoom out again. Big picture. What does a breakthrough like Chai-2 really mean for medicine, for how we develop drugs? It feels like more than just an improvement.
Oh, absolutely. It feels like a genuine paradigm shift. The most immediate impact is just speed and cost. Cutting discovery time from years to weeks changes the economics and pace of drug development entirely.
Getting drugs to patients faster, potentially cheaper. And what about those undruggable targets we talked about?
This is where it could be truly transformative. That ability to design binders for specific epitopes with atomic precision, it gives us a rational way to finally tackle targets that were just too complex or lacked obvious pockets for traditional methods. It opens up new therapeutic possibilities.
Like having a master key designer instead of just trying random keys. And does it help with making better drugs too, not just finding new ones, like those advanced antibody formats? Definitely. Think about antibody-drug conjugates, ADCs, that deliver chemo directly to tumors, or biparatopic antibodies that grab a target in two places. Designing the precise binding arms for these complex molecules becomes much easier if you can generate specific binders on demand.
So, more sophisticated therapies become more feasible to engineer.
Right. It streamlines the design of these next-generation biologics.
And you mentioned it's not just limited to antibodies and proteins.
That's another really exciting aspect. Because the AI reasons at the atomic level, the principles can extend to other types of molecules. Macrocycles, peptides, maybe even designing small molecules eventually, or enzymes with new functions. It's about understanding and designing molecular interactions, fundamentally.
So the core change, the real shift,
Yeah.
it's moving away from just searching and screening, hoping to get lucky,
Yeah, that empirical, almost random approach.
towards actually programming the discovery,
Yeah.
designing exactly what you need.
Exactly. It's shifting from stochastic screening to intentional programmable discovery. The ultimate goal, really, is to get to a point where you can computationally design a drug candidate that's basically ready for those critical preclinical tests, the IND-enabling studies, almost entirely on the computer.
Design it right the first time, or close to it.
That's the aspiration, moving from biology as observation and screening to biology as true engineering.
And it sounds like the team behind this Chai Discovery, they're also thinking about using this power responsibly.
Yes, they explicitly mention a commitment to prioritize applications that positively impact health and society and minimize safety risks, which is important with such a powerful technology.
Absolutely. Okay, so let's try and wrap this up. We started with the huge challenge of antibody discovery, slow, expensive, often failing.
A real bottleneck in medicine.
Then we dove into Chai2, this new AI platform from Chai Discovery, doing zero-shot design with its multimodal, atom-level approach.
Achieving really unprecedented success rates in lab validation, like 16% for antibodies on novel targets, 68% for mini-proteins, a 100-fold jump in some cases.
It compresses timelines massively, tackles previously impossible targets, and enables the design of advanced therapeutics. It really seems to be turning drug discovery from a game of chance into a field of precise engineering.
It's a glimpse into a future where we design medicines with intention, based on deep understanding, powered by AI.
Which leads us to that final thought for you, the listener, to ponder. Given this kind of speed, this kind of precision in designing the fundamental molecules of life, how might this change our ability to respond to, say, the next pandemic?
Or design truly personalized drugs for rare genetic diseases.
Or maybe even finally unlock cures for conditions like Alzheimer's or complex cancers that have stumped us for so long. The potential seems immense. With tools like Chai2, are we finally getting the keys to master biology at the molecular level? Something to think about.